Hello everyone, welcome back to my channel Pari's tutorial. I'm back with one more interesting video. So let's get into the class. Good morning one and all. Welcome to my channel Pari's tutorial. Welcome to one more class. Today I want to talk about biomolecules which are very important for our syllabus to cover in PG course, for the entrance in PG course. Okay, so in the last class we have already discussed about how to attempt the exam, how to get registered for the COET PG exam, and what is the notification, how to go for the website and all, right? So today in this class let's start our life sciences as soon as possible. So the topic that I have chosen today is about biomolecules. So because you all be aware of nucleic acids, which is one of the important biomolecules. So in cell biology, in molecular biology, you will come across a lot about DNA. So I thought let's start with DNA and its structure, its arrangement of nitrogenous bases, purines, pyrimidines, structurally, and how are they arranged, what are the double bonds, what are the bonds that are present, what are the linkages, how the sugar and phosphates backbone is formed for a DNA. Okay, let's all discuss in this class about biomolecules. Okay, so let's start with um, so let's start with biomolecules today. Okay, so what are biomolecules? So biomolecules are considered to be the chemical compounds that are found in the living organism. They are the building blocks of life. They are mainly composed of mainly hydrogen, oxygen, nitrogen, sulfur, and phosphorus. So these biomolecules can be categorized into carbohydrates, proteins, nucleic acids, and lipids. So let us discuss now, as because nucleic acids being very important for our subject regarding molecular biology, cell biology, and everything, I want to concentrate mainly on nucleic acids. So these carbohydrates and lipids I will discuss later. Okay, and these proteins will come across during protein translation, where the proteins are generated in a cell. I will detailedly explain that in molecular biology. We have got a separate class for it. I will tell you in detail. So let's start our class with DNA. So what is DNA? DNA, you all know, it's a deoxyribonucleic acid, and RNA is a ribonucleic acid. Both are nucleic acids, but only the difference between DNA and RNA is the DNA has a ribose without oxy group. Okay, and this contains ribose sugar with OH group in it. So should be well aware of the structures also. Okay, see, I will tell you in detail about the structure of DNA and RNA. Okay. Yeah. So, what happens is, so let's talk about DNA now. So I told you DNA is a ribose sugar with which is a pentose. So this is your right. This is your this is your first carbon, um, second carbon, third carbon, fourth, and this is your fifth position. Okay, to this ribose sugar, I mean to this fifth carbon, you have a phosphate group attached. So this is your phosphate group, and this is your ribose sugar. As I am talking about a DNA, I want to talk, say it's a deoxyribose, because in the second position of this you have right, this is your DNA, right? In the second position you have H, instead of RNA you have OH. So as O group is lacking in the ribose part of DNA, it's called as deoxyribose. Okay, so please be aware that the fifth position is meant for the attachment of phosphate group to form the phosphate sugar link here, and for the first carbon compound there is an attachment of your nitrogenous base. Okay, attachment of nitrogenous base. Okay, so now what are nitrogenous bases? Nitrogenous bases are the organic bases which contains a nitrogen group in it. Okay, so these nitrogen bases are mainly of two types. Okay, they are purines and pyrimidines. Now let us talk how they are arranged in a DNA molecule. Okay, so you all know that the DNA is a double stranded DNA. Structurally it has got 5' side, 3' side, and 3' side and 5' side placed in anti-parallel direction. Okay, so you all know that the number of purines, like I'll talk about them in structurally also in this class. Okay, the purines and pyrimidines, the sugar phosphate group, ribose sugar, and the nucleotide, all these three nitrogen base, sorry, phosphate group, ribose sugar, and nitrogen base, they form a nucleotide. Okay, so each and every strand is made of polydeoxyribonucleotides for DNA, right? They have many nucleotides. Okay, and ribose sugar plus nitrogen base is called nucleoside. Okay, today in this class let's talk about what is nucleotide. Okay, so there are different types of nucleotides. What are the classes, what are their structures, how are they arranged? Let's talk now with a detailed description of their structures. Okay, so this ribose and this phosphate sugar form the backbone of the DNA. Okay, nitrogen bases are mainly of two types. They are purines and pyrimidines. Okay, structurally we have to be aware of their structure. Like, what are purines? Like adenine and guanine are called purines. And what are the pyrimidines? Cytosine and thymine in DNA are called pyrimidines, and uracil is present in RNA. And I don't want to talk about that now. I am mainly concentrating on DNA. Now let's start about the structure of DNA. Sorry, the structure of the nucleotides, purines and pyrimidines. How are they arranged, and how the structure resembles? Okay, let's talk about pyrimidines. So pyrimidines resemble the structure of benzene. So you should be knowing a benzene structure, which is C6H6, right? So benzene, you might be knowing the structure of benzene also in your chemistry classes. Okay, so this is your structure of benzene with the valency. You know that carbon got a valency of 4. You have to balance it with hydrogen, right? So double bonded, and you have double bonds also in benzene. So in the same way, pyrimidine resembles benzene structure, but instead you got a nitrogen group in 1 and 3rd position of the carbon. That is the first position, this is 2nd position, this is 3rd position, this is 4th position, this is 5th position, and this is 6th carbon. Okay, in pyrimidine, instead of carbon you have a nitrogen in 1st group and in the 3rd position. See, based on it I am drawing the structure of the pyrimidine ring. Okay, it has got only one ring. Purines got two carbon nitrogen rings, but pyrimidine got only one carbon nitrogen ring. It almost resembles benzene structure, right? So it should be again balanced with double bond. Being nitrogen having a valency of 3 and carbon having a valency of 4, you have to balance it with hydrogens. Okay, so this is your basic pyrimidine ring. So what are the pyrimidines that you have? The first one to be the cytosine, right? The cytosine has got an amino group in the 6th carbon, right? So, and also this is called the cytosine, right? Yeah, this is the amino group that you got, and in the 2nd position you got a keto group. Okay, that's called cytosine. So the other pyrimidine that we have to discuss is about thymine. What is thymine? So thymine is also a pyrimidine with a 1 CN ring. the same structure, but it has got a methyl group in 5th carbon position. Okay, thymine also got methyl group in 5th carbon along with a keto group on the 2nd carbon. See, it also got a keto group on the 2nd carbon of thymine. So this is your thymine structure. The main thing resembles here with RNA is absence of your methyl group. So RNA doesn't have a methyl group attached to 2nd position. So you call it as a uracil. Okay. So this is the structure of thymine and that is the structure of cytosine. So these are the pyrimidanes that are present in the polynucleotide strand of a DNA molecule. Okay, now let's talk about the purines. How are the rings attached and how are they bound to each other on one side of the DNA with the other strands of the DNA. So the purines are having 2 CN rings, right? So one pyrimidine ring is the basic ring and attached with it you have got one more CN ring. So this is a common pyrimidine ring that we already know, and to that you have an extra CN ring. Okay, this is the basic purine ring. So you have to balance it again with double bond and valency has to be satisfied, right? So what are the purines that we have? They are adenine and guanine. Okay, so adenine and guanine. In adenine, so adenine has an amino group in the 6th position, right? So this is what you call, let's numbering 1, 2, 3, 4th carbon, 5th carbon and your 6th carbon, 7th, 8th and 9th. Okay, this is your adenine I'm talking about. Next talk about guanine. For guanine, draw the same way. This pyrimidine ring is common, okay, but for guanine you have a carboxy group here and an amino group here. Okay, you can do it with a double bond also. Okay, see, right? So you have to balance with a double bond. So nitrogen has a valency of 3, so I'm balancing it out with hydrogen. Okay, done. So this is the structure of adenine and this is the structure of guanine. So you are well aware of it, right? So okay, now let's go for the desktop. Yes. So these are the nucleic acids that I'm talking about. See, that I have already discussed: purine, pyrimidine and etc. Okay. Now, so this is the important thing that I want to discuss. Okay, see, this is the 5' end of phosphate group and this is a 3' end. Okay, and this is the OH 3' end of ribose sugar. Okay, this is a 3'. This forms the phosphodiester linkages. So sugar and phosphate backbone is mainly of the 3' end of ribose sugar with the 5' end of the phosphate group. Okay, see, this is the linkage. Okay, they are in anti-parallel direction. Okay, see, and the first carbon of ribose sugar is where the nitrogenous base is attached. In the same way, the first carbon of this ribose sugar you have thymine attached. The first carbon of this ribose sugar you have a guanine attached. This is the first carbon, you have adenine attached. So the first position you have nitrogenous bases attached and the 3' you have your sugar and phosphate backbone. Okay, so this is the triple bond that is formed between cytosine and guanine, and it is a double bond between adenine and thymine. So this is how a DNA is structurally linked in each strand, how they are properly arranged. So I thought it would be important for me to discuss about a topic, a question that was being asked last time. Okay, that question was if the GC content in one DNA is this much, what is the percentage of other nucleotides? So let's discuss about that now. Okay, so the question was, if GC content in DNA is 60 percentage, what is the percentage of all the bases present in DNA? So for this you have to follow one important rule called Chargaff's rule. So you all know that. So let's draw a linear form of DNA. Example, let's say DNA. Okay, I'm just making it as simple as possible. Okay, so this is the two strands of DNA, and for example, this is A, this should be T. If it is G, this should be C. If it is C, this should be G. If it is T, this should be A. So it is like that only. So I'm just drawing it rough. So now they told us the GC content given is 60%. If this is the 60%, what would be the other bases? So it's quite easy. So all purines on one strand are equal to the pyrimidines on the other strand, right? So if G and C are 60%, guanine should be 30%, and cytosine must be 30% as per Chargaff's rule. In the same way, we all know that the out of 100%, if 60% is accompanied by GC, the remaining 40 would be accompanied by 80. And the same rule is followed again. 40% should be equally distributed because adenine is a purine and thymine is a pyrimidine. Right, so adenine should be of 20% and thymine should be of 20%. So this is the important question that they have asked. In the same way, if Chargaff's rule, okay. So the answer for this question is G and C are this 30 percentage, and A and T are 20 percentage. Okay. So this is a question that I thought I should discuss. This is a right moment for me. So then I thought to discuss about this topic. Okay, that's why I'm telling you that it has come, the time has come, so I thought I can share this with you. Okay, so now let's go for the structure of DNA. So in the structure of DNA, I already told you this is the sugar phosphate background. These are the AT links, these are the nitrogen bases, and this is the approximate thing that I've read about. So there is a diameter of the DNA is generally 20 angstroms, and the pitch, the length required for one complete turn is 34. Okay, in each DNA strand the bases occur. It's like if you take one pitch of DNA, a number of bases per one pitch is 10 base pairs. Okay, it's a rough amount, it's an approximate amount. Just not completely it's a rough amount that you should get an idea how many strands are there per one turn of DNA. Okay, so each of two DNA, so it's a multimer of deoxyribonucleotides known as polynucleotide. They are known, then you all know that it's an anti-parallel direction. Phospholinkages are anti-parallel direction. Okay, now I already told you about the Chargaff rule. Okay, I already told you that these are the linkages that are present in between the bases. Okay, so like this video, please like and share and subscribe my channel. Please encourage me for the further videos. Thank you so much. See you in the next class. Bye.